In the summer of 1928, penicillin entered human history not through a planned experiment, but through a moment of chance noticed by a prepared mind. The world before penicillin. At the start of the 20th century, bacterial infections were among the leading causes of death. Pneumonia, sepsis, syphilis, and infected wounds routinely killed otherwise healthy people. Surgeons feared infection more than the knife. Treatments existed, antiseptics like carbolic acid, but they often damaged human tissue as much as bacteria. The idea of a drug that could selectively kill microbes inside the body was largely aspirational. Alexander Fleming and the accidental observation. Alexander Fleming, a Scottish bacteriologist working at St. Mary's Hospital in London, was studying Staphylococcus bacteria. Fleming was known among colleagues as brilliant but untidy. In late August 1928, he left for a holiday, leaving petri dishes of bacteria stacked on his bench. When he returned in early September, Fleming began sorting through the plates before discarding them. On one dish, he noticed something unusual. A patch of mold had grown, and around it the bacteria had been destroyed. Where the mold grew, the Staphylococcus had dissolved, leaving a clear zone. Many researchers might have thrown the contaminated dish away. Fleming did not. He identified the mold as belonging to the genus Penicillium, likely Penicillium notatum. Fleming concluded that the mold produced a substance capable of killing bacteria without harming animal cells. He named this substance penicillin. Early research and initial limitations. Fleming published his findings in 1929 in the British Journal of Experimental Pathology. He demonstrated that penicillin was effective against many harmful bacteria, including those responsible for scarlet fever, pneumonia, and meningitis. However, penicillin was unstable and extremely difficult to isolate. Fleming lacked the chemical expertise and resources to purify it or produce it in large quantities. As a result, his discovery attracted limited attention. For nearly a decade, penicillin remained a scientific curiosity rather than a medical breakthrough. Revival at Oxford. In 1938, at the University of Oxford, pathologist Howard Florey and biochemist Ernst Boris Chain revisited Fleming's neglected paper. Chain was intrigued by penicillin's potential, and Florey assembled a multidisciplinary team to solve the problems Fleming couldn't. Using improved biochemical techniques, the Oxford team succeeded in extracting and concentrating penicillin. In 1940, they tested it on mice infected with deadly bacteria. The treated mice survived. The untreated ones died. First human trials. In 1941, penicillin was administered to its first human patient, Albert Alexander, a British policeman suffering from a severe bacterial infection. His condition dramatically improved after receiving penicillin, but supplies ran out before the infection could be fully eliminated. Alexander died shortly thereafter, underscoring both penicillin's power and the urgent need for mass production. Wartime mass production. World War II transformed penicillin from a laboratory experiment into a global priority. Britain, under heavy bombing, lacked industrial capacity, so Florey and colleagues turned to the United States. American pharmaceutical companies, supported by government funding, developed methods to mass-produce penicillin using deep tank fermentation. A more productive mold strain was discovered on a cantaloupe in a Peoria, Illinois market, further evidence that chance continued to shape penicillin's story. By D-Day in 1944, penicillin was available in sufficient quantities to treat Allied soldiers. It dramatically reduced deaths from wound infections and sexually transmitted diseases. Recognition and legacy. In 1945, Alexander Fleming, Howard Florey, and Ernst Chain jointly received the Nobel Prize in Physiology or Medicine. Fleming famously warned in his Nobel lecture that misuse of antibiotics could lead to resistant bacteria, a prediction that proved prescient. Penicillin marked the beginning of the antibiotic era. It transformed medicine, made complex surgeries safer, extended life expectancy, and reshaped humanity's relationship with infectious disease. What began as a forgotten petri dish became one of the most consequential discoveries in human history, not because it was planned, but because someone noticed something unexpected and understood its significance.